Hey, let's go ahead and get started. Good afternoon, good evening, good morning, wherever you are. So it's a pleasure to get started with the IB Industry Connections webinar series. But prior to that, I just wanted to tell a little bit about the Institute of Biological Engineering. I have the privilege to serve as the president for this year. So the Institute of Biological Engineering was established in 1995 to encourage inquiry, application, and interest in biological engineering in the broadest and most liberal manner, and to promote the professional development of its members. So since its inception, IB has provided engineers, scientists, and students a forum to discuss, debate, and advance the field of biological engineering. So it's a science-based, application-independent discipline that is aligned with the perspective and foundation of biology. I encourage each one of you to visit our website to learn more about the Institute and a lot that's been happening over many, many years. And so today, we are starting the IB Industry Connections webinar series. We're excited to have Dr. Nate Tedford kick off this webinar series. And he's joining us from Boston, where he's the head of the Foundry at Ginkgo Bioworks. He has over 20 years of experience in building technology platforms in the fields of synthetic biology, systems biology, gene therapy, protein engineering, and medical diagnostics in both academia and industry. He's an expert in metabolomics and proteomics, and has applied his approaches to understanding diverse and complex biological systems. He's grown a variety of teams and capabilities in the Foundry, which is the company's highly automated factory for cell programming and high throughput design, build, test, learn paradigms to successfully apply biologically engineered solutions to a number of industries and applications. Nate received his BS in biomedical engineering with a focus in chemical engineering from Johns Hopkins and his PhD in biological engineering from MIT. So today he's going to provide an overview of Ginkgo Bioworks and discuss some specific examples of where synthetic biology has been successfully applied in market verticals, including agriculture, food, materials, pharmaceuticals, and industrial biotechnology overall. So I'm extremely excited that Nate is joining us today. So Nate, the floor is yours.
Thank you very much. Can I share my screen, please? Great. Yeah, first of all, I'm honored to get to take part in this, and thank you very much for having me. Great respect for the IBE. I've had many mentors, colleagues, friends. Right now, my wife Maxine. So I'm going to give very honest feedback on how this talk is received. But very much looking forward to talking to you about what we do at Ginkgo. I thought I'd give you a bit of an overview on the company and how Ginkgo's grown over the last 14 years. Then talk about some of the areas that we work in more generally, dig into a little bit of our technology stack, and then give you some case studies where we've applied our platform for different industrial applications. We recently did take the company public, and so I have to show this quick disclaimer, but no harm done there. Let's dig into it. So as I mentioned, Ginkgo is 14 years old. Founded in Boston in the summer of 2008 by 5 folks from MIT, 1 professor, Tom Knight, and 4 PhD students who I think may have been members of the IBA at the time, and their advisors were as well. So, connection there. We had a very non-traditional start, I would say. So did not raise any venture capital or other institutional capital for nearly 7.5 years of our existence, but rather sort of built the company in a bootstrapped way, raising various government grants and collaborating with different academic labs to build out the first versions of our platform. I joined the founding team when we were 8 people, nearly 11 years ago, and feel very fortunate that I got to see that phase of the company's life cycle before we then became very fortunate to raise a number of private funding rounds, Series A through E, after taking a bit of a risk and kind of diverting from the traditional Boston biotech venture capital scene, and rather enrolling in a program in Silicon Valley called Y Combinator. This gave us, sort of exposure to mentors and investors who thought about both tech and biotech. And I think it really helped to color how we built our platform that I'll tell you about today. As of now, we're approaching around 1,000 people at the company. And as you'll see in sort of the themes that I cover, we don't have nearly as many robots as people, but they do a heck of a lot more work than our people in many ways. And so we'll talk about how we apply different flavors of automation to genetic engineering and cell programming. As Raj said, so we're headquartered in Boston, have around 300,000 square feet in our headquarters in the seaport right on Boston Harbor. Over the last 3 years, have also been very fortunate to acquire a number of companies, both in the US and in Europe. And so we now have a presence in California, in Switzerland, in the Netherlands. And so, learning how to become a multinational organization as well, and bring on a lot of smart and talented people in from around the world. As I mentioned with my disclaimer, we were very lucky to get to take the company public last year. Definitely a really exciting experience, and that's let us sort of deploy capital in interesting times right now, as you'll appreciate, with the state of the economy, and hoping that many good biotech companies make it through these times as new companies come out of your labs and others. But in general, feeling very well capitalized to continue to invest in our platform, to do sort of front end R&D for a number of partners that I'll touch on. At the sort of core of our thesis, we want to sort of empower our partners to deploy different kinds of products to the market. So we don't have any intention to vertically integrate, if you will, but rather we invest our capital and resources into our front-end R&D platform that helps us accelerate the engineering of biology. Our core mission is to continue to work to make biology easier to engineer by doing that and approaching biological design at scale, adding a nice balance of sort of automated, high throughput, rational arrayed design and test in synthetic biology, while also balancing that with a number of ultra high throughput and multiplex methods to improve cells, pathways, and proteins to produce a variety of products. And in general, we like to think like good engineers operating on design, build, test cycles to be able to prototype pathways, proteins, and again, the entire genotype of a cell that's going to have a function that a partner of ours wants, to be able to translate that into a successfully engineered cell, and just as importantly, a viable manufacturing process that can be deployed at scale in the field, so that we can help our partners make enough of the material they need and do that at the cost targets that they require. And so you'll see sort of flavors of that along the way. I think one thing that I'm particularly excited about, and not to say that human health isn't important. Obviously, our field contributes significantly to pharma and biotech, and in general, just advancing therapeutics and understanding of physiology and human health in general. And so obviously, we know that there is a lot of attention for these good reasons in pharma and biotech, but at Ginkgo, we really try to think more broadly, where we are looking at biology itself as an engineering substrate, to be able to use an engineered cell to be able to do something of import in a variety of different fields, not just making drugs. Frankly, I'll say when we started out, and as I said, with such a non-traditional start, we couldn't compete in the pharma market because we didn't have the funding or the time to take the risk of staying on an FDA timeline without either getting gobbled up or failing. And so rather, took more of a metered approach to building out our our Foundry, our automated platform that I lead at the company, to be able to get better and better at carrying out different unit operations to discover genes and enzymes, engineer cells, and then start learning how we could interface to different industries and application areas. And so you see some larger examples here that we've been working on over the last 5 to 6 years, both directly with partners externally and sort of arm's length deals, with internal research and development, and with operating companies that our investors are excited to help us start to use our platform and build the expertise that it takes to deploy a product to the field. So quite a bit of work over that time in different environmental applications, and that's only increasing now. A distinct focus within a few of our operating companies, as well as some recent exciting mergers and acquisitions that we've announced. We're taking on the Ag Biologics department from Bayer, as well as our partner company Join, to build a bit of a super department within Ginkgo that's working on ag biologicals. And then also thinking about the general consumer facing products, thinking about how we can take valuable molecules for flavors and fragrances, different plant extracts that may have a pharmaceutical application or may have more of like a product facing application in a consumer product, and determining how we can learn from the genomics and biochemistry of what's happening in a plant cell and translate that to a more fungible, industrially friendly microbe like a yeast, and be able to scale up production of that product. I'm going to monitor the chat here. Okay, great. Okay, so Raj, you'll field questions for me if anything comes up. I should have said that. Please stop me at any time. Happy to answer questions along the way. Okay, to that point where we don't intend to vertically integrate, we sort of like sitting in this like middle layer where we're aiming to build this platform that not only interacts with commercial partners in these different markets, but also we try to sort of pull in and integrate different technology providers. So, how can we better collaborate strategically with folks who are making liquid handling robotics or different flavors of laboratory automation? What are the cutting edge approaches that are coming from new companies trying to commercialize products that give you different analytical capabilities to characterize the cells that we're building? And so, I kind of find both sides equally exciting, but, sort of selfishly, I like being that technology integrator who gets to see how everything kind of fits together across the design, build, test, and grow cycle in our cell factory. Over the last few years, I mean, obviously challenging times during the pandemic, but again, we continue to take that inward focus to not only lean into COVID response and try to be helpful during the pandemic, but also grow the company in a variety of ways. One thing that has changed for us as Ginkgo has grown is that we've been able to acquire different technologies, and to that end, have been able to sort of balance bringing in what I'll explain as code base assets, or sort of interesting and valuable genomics data, different kinds of cell types that will have different advantages for making different kinds of target molecules or products, and then technology integrations that let us make our platform better. Those range from sort of technology and capability and being able to write and manipulate DNA, to being able to handle populations of diverse cells at massive scale, to more sort of hardware and technology stack plays, being able to do online NMR on fermentation systems, or being able to bring about more sophisticated automation. And so definitely been a learning experience for me and the team to see how you grow a company both organically, internally by hiring great people and building out your tech, as well as bringing on new teams and having them feel integrated and welcome on the platform. So quite a lot of progress there, and something that we think about a lot these days. Just to touch a little bit more specifically on the kinds of projects that we run on our platform. So today, I think we have around, we're approaching 100 active products in active projects. Now this slide's a little bit outdated. You know, interestingly and challengingly, in over 50 different species. So, you know, different microbial cell types, different fungi that, you know, may have not been so friendly to work with in the lab at one point, but we find ways to develop tools for them and domesticate them to be able to make interesting and complex products. And now increasingly so also we're working in mammalian systems for 4 different biopharma applications. So, you know, don't want to completely ignore pharma. Obviously, it's an important and exciting space and something that we're growing our platform to aim towards. But as you see here, you know, sort of in that therapeutic area, kind of a mix of sort of antibody discovery or improvement, antibiotics discovery. So looking at natural products pathways in soil bacteria, for instance, that may give you, you know, the ability to get around concerns in antibiotic resistance and give you new classes of antibiotics. We look at, you know, both industrial processing enzymes for pharma as well as therapeutic enzymes. And then I'll touch on a case study where we're working with various partners on what we call living therapeutics, either for metabolic syndromes or sort of microbiome related applications, or increasingly so looking at gene and cell therapies, trying to, you know, apply our sort of systematic way of biological design and thinking and optimizing our receptor or cell design to provide a more effective therapeutic. And then in the non-therapeutic area, again, you know, a lot of learning from both the industrial chemicals industry, and, you know, also learning from sort of the first and second wave, I think it's fair to say, of industrial biotech, where there were some bio-based solutions where, you know, folks were learning how to use either a naturally occurring or engineered cell to make a product of interest at scale. But, you know, again, scanning from different industrial applications through to ag, food, and different sort of products for formulations as such. So for us, the way that we organize the technical platform at Ginkgo, so we have sort of 2 primary components. So we have our Foundry and our code base. We will interface with our partners to know what specifications they need for an engineered cell that we will program and improve for them to be able to make either a small molecule product, a protein product, or the cell itself as the product. And all that physical work happens on our Foundry program. And so within the Foundry over the last decade, we've really been heavily investing in a mix of off-the-shelf software and hardware and custom software and hardware to be able to bring to bear sort of high throughput workflows and capabilities in every aspect of sort of early stage discovery, cell improvement, and then process optimization and deployment. On the sort of bottom half of this flywheel, as we perform that work, we're very mindful to generate data and do as much with that data as we can. You know, we certainly can't profess to be the perfect designers of biology yet. You're working with very complex systems with interactions where when you're pulling heterologous genes from a variety of sources to gain the biochemical activities you need in your programmed cell, you don't always know what's going to happen when, you know, an enzyme you express that wasn't in the cell before is now interacting with native metabolism. You know, it may recognize other substrates. You know, you're obviously making cells typically unhappy when you're doing these things. You know, they want to find their carbon source and make copies of themselves, and instead you're trying to divert all of that metabolic flux into something that they typically don't want to make. And so you're always fighting a bit of an uphill battle in convincing the cell to do that in a way that works in a sort of stable and robust manner over the time of a, you know, many day to many week in some cases fermentation or bioprocess. And so, you know, we take the data of our good designs, but equally important of our bad designs, and we feed that into what we call our code base. And so over the years, we have been able to build out, you know, different forms of what we call our code base. So, you know, we have different tools. So when we get familiar with a given kind of microbe to make us a certain class of product, we're often collecting knowledge in terms of what kinds of biological parts can help us more effectively drive expression of the genes that we need to build a pathway and give us biochemical function that's desired. And so those sort of biological tools come as a way of like a collection of regulatory elements, better gene editing systems, better sensors or tools that will tell you, hey, the cell is in a state of stress, or I'm seeing metabolic flux that wasn't predicted. I'm sensing these molecules. How can I further improve my pathway dynamics in a cell design? We also learn a lot about how we code our DNA in a given species. As many of you appreciate, we all have A's, C's, T's, and G's, but whether you're some esoteric soil microbe or a tree or a dog, you're going to use different rules and different codings in terms of the codon frequency you use, the regulatory schemes you use to drive effective gene expression. And so one thing that we really value is the fact that being able to generate data at scale lets us sort of recode and refactor that sort of playbook and how you're going to write a series of genes and use a series of regulatory elements to make something work if you're sourcing genes from a variety of plant sources or esoteric microbes and putting them into a more tractable industrial host. And then finally, as I touched on a bit, we've worked with over 50 hosts. Just that base know-how and knowing how to grow and scale different kinds of organisms is also important. I think for us, it was probably partly because of our non-traditional start, where we were so poor and so scrappy that we had to take any project that we could get our hands on early on, that it really forced us to think about how to add some flexibility to our platform to be able to bring on non-canonical hosts. And so as you look across the industrial biotech and synbio industry, oftentimes you'll see that a company will have like a specialty. There'll be the E. coli company or the Saccharomyces company. And so, I think there's a lot of power in building out that diversity in our platform where we can pick the right host for the job, depending on, what our partner needs. Over the years, we've rolled out different versions of our Foundry. So we call these our Bioworks. Here's an example of our first 5 moving from sort of, semi-manual work to sort of translate processes to walk-up lab automation. Then, sort of halfway through the progression to date, it's really started to sort of lock down processes that we knew could scale. And, doing that in a more industrialized way, but not completely. Fast forward to sort of the latter half of our development arc thus far, and we've really invested heavily in a core sort of software and hardware capability. So like I told you, that Ginkgo's approaching 1,000 people. About 190 of those people are either software developers or automation engineers. And so that looks pretty non-traditional even to like a cutting edge biotech. And so, that's where we really kind of strike that balance between being like a tech company and a biotech company. But what it allows us to do is think about adding practices in more sophisticated integration of automated systems, thinking like a process engineer or an industrial engineer to think about how we can translate different lab protocols and basically turn those into fully walk-away automated workflows that happen on robotics. What this lets you do is start to think differently about the scale of design that you can approach in an experiment. So for instance, when we're trying to discover a new enzyme that will carry out, the 5th step of biochemistry in the pathway that we think is going to be the best way to make a small molecule product for a customer, we will oftentimes source many thousands of enzymes. And we can only do that with this robotically assisted automation where, a work cell that we'll build that I'll talk about in more detail a bit later is, oftentimes able to do the work of 6 to 10 people in a day and really change how you can source biological diversity. And that just like right off the bat, leaning on the power of nature, in terms of how you can think about different starting points for genes that can give you what you need in your cell design is quite powerful. And so, common theme that you'll see there, but, some of this is brute force, some of it is, intentionally sort of targeted through computational modeling and other guided approaches, but, sort of going big in biological design and then being able to test and generate data is a very powerful combination. What we have aimed to do in sort of, maturing and refining this sort of tech stack is to stay on a trajectory that we call Knight's Law after our founder, Tom, where, we're trying to kind of like hold ourselves to output metric and performance criteria similar to Moore's Law. We have all seen in our field, the great advances in, sort of improvements in the cost of sequencing DNA and synthesizing DNA. Our sort of core thesis was to keep building on that. And if we could apply those same sort of technology advancements, we could get to a point where we could even do that in more complex end-to-end workflows. So for instance, over the last 7 years now at Ginkgo, year on end, we have 3x the output of strain designs and tests that we generate while trying to drop our costs in half every year. And we can really only do that by, sort of scaling and professionalizing our workflows with laboratory automation, as well as smarter and smarter software and computational analysis tools to be able to track what we're doing and translate workflows into robot speak, and then be able to capture all of the data and metadata that you need to make sure that you're running your work correctly. And so, something that we really try to stay on. I mentioned, that we try to think like engineers, and for those of you that work in the synthetic biology space or sort of proximal fields, the concept of design, build, test, and ferment or bioprocess is not new. Many great academic labs and companies in SynBio very rightfully have added the learn layer as well. So sometimes you kind of forget about the ferment and you add, it's more of a DBTL cycle, which, well, my slide doesn't show this, is extremely important. And we try to add that L actually at every single stage of this to figure out, what is working and how we are generating better cell designs and getting the data that we want to expect. I mentioned the technology and software layer and how we really had to make that commitment to build things. I think especially 10 years ago when we were looking at this, there was really no off-the-shelf solution for an end-to-end stack that would carry out every piece of that complex design, build, test workflow. And so, something that we were intentional about saying, well, I guess we have to build it. And so, it took a long time to get it to a place where you could say, hey, yeah, we really do feel 50x or 200x more efficient. Frankly, in the beginning, we felt less efficient. We could have hired a bunch of smart PhD students and postdocs and probably done a better job, but kind of staying on that mission to lock down processes and learn how to systematize them and run good process on the platform has been very important to help us scale. And so across this sort of design, build, test, and ferment ecosystem, as I mentioned before, we've kind of made bets on both externally available partnered technologies and stuff that we build in-house. On the front end of the platform and design, we have made use of publicly available algorithms, used to source genetic code, to discover enzymes, to build and balance pathways, to understand sequencing data when you're doing discovery work or understanding the overall genotype of a cell you're working in. Also use that to guide, approaches in protein engineering and strain selection and evolution to find new tools and improve cells and proteins in new ways. On the build front, we are the largest customer in the world of commercial synthetic DNA. So, we have now re-upped on our second 1 billion base pair deal with Twist Biosciences in the Bay Area. We're also very lucky to get to acquire Twist's biggest competitor 5.5 years ago, a company called Gen9 that was located in Cambridge here in the Boston area. Gen9 moved over to Ginkgo, became a team in the Foundry. and really gives us the ability to build all the hard stuff in-house that none of the commercial vendors will build for you. So we can build, really long sequences, about 2.5 times longer than any commercial offering that's available, as well as deal with, kind of complex or frankly ugly sequences. So, repeat sequences, higher low GC content, and the like. And so it's really nice to have that balance of external and internal DNA synthesis. What that lets us do is approach gene length or multi-gene length synthesis at scale. So, again, it is routine for us to order a library of 2,000 to 5,000 genes when we're trying to improve the step of a pathway. We also, again, as I mentioned briefly, think about how to complement sort of rational design in a RAID where you have sort of one genetic design per well of a multi-well plate going into your testing workflow with, sort of more clever or elegant pooled approaches. So how can you sort of multiplex your design by doing combinatorial assemblies or introducing diversity via various mutagenesis schemes to be able to generate new starting points and hypotheses. Once we've used those different build functions to program our cells, they go into a variety of different test beds, ranging from sort of targeted high throughput screening to more comprehensive metabolomics, transcriptomics, proteomics, do quite a bit of sequencing in-house, as well as add kind of a balance of sort of slow and comprehensive analytics on the back end and know that we're making our molecules of interest versus, at the start of a project doing very high throughput work. And then finally, at the end of the day, you need to get your best cell designs into a fermenter and know how is that cell design performing? Am I hitting my productivity or tighter targets? If my customer needs 12.5 grams per liter on day 4 to make an economically viable process, I need to make sure that I'm replicating that at different scales, first in our lab in the Foundry, but then in pilot scale facilities where we'll go from sort of quarter liter or liter scale fermenters up to 50 liter, 500 liter, 5,000 liter to say, okay, I feel confident that I can deploy to full scale. And so, the learnings that we've had over the years of learning how to stay as small and nimble as possible in the formats you're using, but building data models that let you predict what winning cell design will do well at scale is crucially important, and something that we've interfaced well off the back end of our platform. A little bit of talking about more specifics in our tech stacks, and then I'll go into some case studies at the end. So, one thing that I think differentiates the way that we approach the design build test cycle in SynBio is we've kind of taken a married approach of what you might see in a high throughput screening facility in a big pharma company, versus, learning how to build more flexible automation that isn't just screening, a library of compounds against a P450, but rather having hardware and systems that are able to use integrated robotic arms that can link anywhere between 10 and 30 pieces of hardware, and being able to run workflows that let you sort of build on translating protocols of increasing complexity and increasing size. And so both our in-house automation and software teams and our external partners like Hi-Res Bio, our newly acquired or soon to be acquired company Zymergen, who's also worked in this, is going to complement the stack as well. But in general, making investments where I feel comfortable paying $2 million for this Ironman work cell because I know that it's going to be able to run 30 different protocols for me, being able to very flexibly switch configurations within a couple of hours and running every day, batches of screens that can, get me data across very complex workflows so that I can get hundreds of thousands of data points, whether it be running whole cell-based assays, detailed time courses, measuring cell viabilities, measuring enzyme activities, and measuring different kinds of protein binding. That's the kind of work that we can translate onto this integrated robotics and sort of run our tests at scale. And you'll see in the examples where I think that's very powerful because it gives you, the ability to look at a broader diversity in your biological design that you're interrogating. The most sort of advanced facility that we have built thus far, Bioworks 5, you see one of the 7 lines in that facility that is actually being run by magnetic rails. So this is like a biotechnologist's nerdy train set of sorts, where you're able to use magnetic force to levitate plate carriers and then send them around these tracks where they can be addressed by these, green robotic arms. These multi-axis arms are something that, you're more used to seeing in like an automotive factory. But what these can do is vastly increase the throughput with which we can move samples and reagents and carriers, so that you can, really in many cases, 10 to 20x the scale of what you could do on those systems that I was just showing you that we've integrated around our labs. We built this facility in the first year of the pandemic to be able to do next generation sequencing based diagnostics for COVID. As I had mentioned, decided to actively lean into the pandemic response and do work for a variety of collaborators and on our own. But we built this facility intentionally with the ability to sort of refactor it and take all the learnings over the last 5 years of building out automation to continue to make it flexible still, where on each line we could lock down different kinds of protocols to work in DNA assembly or purification or sequencing, or set up for high throughput screening. This facility cost $50 million to build over 9 months. We had our entire team of, at the time, around 155 automation and software engineers working on it, as well as an additional 120 engineers from a few engineering contracting firms. And so we really brought in expertise from, traditional manufacturing industries and some of the newer players that work with different biotech companies to help in integrations. And so, maybe the closest comp to this facility is an automated chemistry facility that Eli Lilly built in San Diego with the help of some of those contracting firms. It took them 3 years and about half the budget to build, but we did not want to wait because we knew that we didn't know when vaccines were going to come out yet, and we wanted to make a contribution. We've now distributed our testing business around the country, mainly for K through 12 classrooms and for airport surveillance. And so now, as I mentioned, running core cell engineering workflows on there. And so, what we've learned how to do is take all of our sort of process monitoring software and workflow translation software and take what a single work cell was doing and now sort of link it together on this sort of train line that's able to run multiple protocols in concert in a walkaway manner. So when I think about how I staff my sort of specialized teams across the sort of design, build, test cycle, typically in a Foundry space, I'm putting 55 to 60 people in a Bioworks. In this space with this, very, very refined and sort of interconnected automation, we can run this entire facility with 12 people. And we had designed the facility to spec to be able to do 100,000 diagnostic tests a day. We have now chopped it up into a few different lines to run our cell engineering workflows, and we're able to run around 2,000 to 10,000 samples in batches for next-gen sequencing, for example, and have those batches running daily, if not twice a day. So we really get a scale economic there, where now we're making sequencing so cheap that we can sequence everything that we make. And really have like a nice robust proofreading pipeline in addition to using sequencing as a discovery tool. We've been a partner at the forefront of working with folks who are making mass spec high throughput, using different acoustic liquid transfer technologies to be able to basically generate sub-nanoliter droplets of your sample and to be ionized and sent to a mass spec. This is transformational because what we find in our field when we're working across the 100 projects that we're working on is that about half the time you can't detect your product of interest with a simple plate reader assay. You actually need a mass spectrometer, and mass spec is inherently slow. LCMS and GCMS runs can range anywhere from 2 minutes to 30 minutes per sample. This system allows us to introduce sample and ionize it and get a peak detection in a second per sample. And so, now we're running like high throughput screening libraries with mass spec and have, worked with our automation team to make robotics that will basically feed this system with plates all day long to be able to run libraries of, 50 to 100,000 cell designs, driving expression of different enzymes. On the sort of ultra high throughput side of things, we do a variety of different kinds of computational and wet lab protein engineering approaches. This is a super exciting time for protein engineering right now. You guys are likely familiar with the advances that like AlphaFold and other computational tools have brought to the field. We're really trying to invest heavily here to be able to sort of think about how we can improve different aspects of proteins. We think about enzymes all the time, as well as different binding proteins. And so knowing that we can make a protein more soluble and expressible, more specific for a substrate, more stable, or both from a mutagenic and also from substrate recognition perspectives, more sensitive. Things that we think about across most of our projects. And so we've developed a number of sequencing coupled based techniques that let us do sort of mutational scanning across an entire protein sequence and then generate back data to help inform those models to be able to improve structure and function for proteins. We also think a lot about biosensors. So if you can essentially evolve a molecular sensor that will recognize a molecule of interest, you can oftentimes couple that recognition with some output, be it a growth advantage or a sequenceable transcriptional output that lets you pool millions of cell designs and have the winner show itself. And so, not for nothing, it is not trivial to make a good, robust biosensor, but when you can do that for a product of interest, you can really take a variety of very elegant pooled selection and evolution schemes to make an engineered cell better at making your product in the context of the process. We have different barcoding strategies that let us do this kind of pool design even across pathways, and I'll talk about that in more detail a bit later. And I'll say our most recent acquisition of a company has brought a really exciting technology to our platform, that being an encapsulation and sorting scheme from a company formerly known as FGen in Switzerland, which has sort of hardware and assay development and selection expertise where they can make a workflow where you can take a population of 10 to the 5th to 10 to the 7th cells, send them through a microfluidic device to capture roughly 1 cell per bead and an alginate bead that is still allowing for the mass transfer you need to have the cell get the food it needs and the oxygen so that it can continue to grow. So you basically get a form of flow cytometry enabled backend where you can amplify your signal by growing hundreds or thousands of cells in each bead, but knowing that that bead has a single genotypic design. And then what you can do is actually throw those beads into a stirred batch or a shake flask, basically putting it in the process that you're going to be manufacturing in, and then taking with some ability to have a readout for the, the performance you need to be able to have that cell go to, showing you the winning design that you can sequence. I'll stop there. I have some case studies that I can point out. I know we're at just past 45. I know this is an hour and a half, but I don't know what the standard format is. If you guys would like Q&A or kind of touching on different things, but I could touch on a few of these and then, or we could pause for a bit and then return to it. I defer to you guys.
Thanks, Nate. I think it will be nice to entertain some questions right now and then maybe go into some case studies as well. So we'll open the floor up for some questions, and then maybe it'll be interesting to look at some of these case studies. Yeah.
Perfect.
Okay, I think I'll go ahead and start off with a question. So I have sort of a biased question to ask in the context of in the context of regenerative medicine, right? We are really focused on cell-based therapies, and so the cells are the products, right? And so I was just wondering, what are some of the things that you're looking at when we are engineering cells or reprogramming cells, especially in biomanufacturing paradigms, to ensure quality control. So basically, for example, like genomic stability is one thing that is a big concern. So I was just wondering if you could address that.
Yeah, great question. So in some ways, well, genomic stability is so important, and it's something that in many places has been ignored because companies who have commercialized either cell lines for production of a protein product are many times afraid to know their entire genome sequence because they're afraid they might be infringing on a competitor, or they don't understand why something is working. I think that's really changing today. So there's a lot of opportunity to improve cells as factories, and mammalian cells in specific. A lot of good work going on across some different labs, looking at how to make the super CHO or hex cell that's going to be a better producer. I think that there's also a lot of opportunity now to think about how to improve gene therapies and cell therapies in general, and not just think about bioprocess and manufacturing, and use both the ability to generate more diversity in biological design, say for a T-cell receptor or a CAR receptor, or to understand what the sort of underlying genome and genotype of the cell is doing to drive performance. And so yeah, I think just being in a world now where you can basically read everything is quite powerful, and I think more people are starting to do it and should do so increasingly.
Thank you, Nate.
Any other?
Yes.
Yeah, I have a question regarding one of the new verticals you say you're getting into is biosecurity. And I wonder if you could tell us what that means. In particular, it seems to me that it would be something challenging to do in isolation, just as one company. And so I wonder if you have partnerships or governmental alliances or mandates to do that.
Yeah, totally. So in the biosecurity front, I'll show part of this case study. So yeah, you can't go at it alone. You need partners. This first part is more in manufacturing. But what we have built out over the last 2.5 years is a separate business unit at Ginkgo called Concentric. Concentric today is a team of around 250 people who are experts in biosurveillance, bioinformatics of viral samples, as well as, just as importantly, understanding the policy in a given country or region and the sort of norms around acceptance of testing, of monitoring. We kind of see, like, thinking specifically about the pandemic, these sort of 3 phases of like mitigate, intercept, and detect, where first you're thinking about like, how do I deal with the problem at hand, and then how can I sort of test and trace my way out of that problem to some extent. And we've seen that work to better or worse outcomes in different places around the world during COVID. After that point, I think, working with partners to actually do more careful and comprehensive biosurveillance is really important. So they're not just doing a targeted diagnostic for the variant of COVID you care about, but having more flexible methods to understand what pathogens might be present in a region. And so, I think that there's a lot of good infrastructure and learnings now built out where you can do wastewater sampling. You see companies like BioBot who have like built out that ability to have that infrastructure. We've partnered with countries in Asia and in Africa and Europe to be able to run that. I mentioned we had distributed our school testing to all the states in green here. Done over 10 million tests to date now. This is only slightly outdated. But more importantly to me, test and trace is important. You want to keep kids at school. You want to make sure that you're tracking outbreaks. But we've taken a sampling of those and essentially developed a whole genome sequencing service where we can have flexible adapters and primers that can not only go after COVID, but many classes of virus or other pathogen. And so, actively working with different countries and different airports. So here's a publication we put out where we've been able to pick up the first instance of some of the new COVID variants that are coming into the country from different routes. knowing, okay, this lineage is coming off of BA4 or BA5, this looks completely new in tracking, but yeah, as I said before, equally important that you're able to work with partners to not just look for a single thing. I think that sequencing is incredibly important there because a lot of the more traditional diagnostics are too targeted. And so you're only looking what you're testing for. And so having that flexibility to more broadly interrogate what is in the environment is powerful.
Thank you.
Okay.
Other questions? I'll ask one more question. So again, it is nice that you had listed the some of the aspects of the UN sustainable goals very early on, right? So, and again, shows some of the social responsibilities that you have, and so, especially in the ag space, how do you ensure that some of the things that you do in your company, especially in the context of cell reprogramming, either contributes to maintenance or hinders biological diversity in different environments?
Yeah, it's a great point. And it's an important challenge in the application area. I think if you talk to certain experts in the field, they'll tell you, oh, well, if you're deploying an engineered cell into the field, you can just make a kill switch. And, that's going to solve your problem. You're not going to have widespread, this newly engineered cell is going to take over an environment. I think that's somewhat irresponsible because we know that things can sort of evolve and escape. And so you have to be very careful to know what other sort of controls and mitigants you can put in place. For many application areas in ag, what we have seen in recent years is that, for better or worse, usually when you're doing heavy engineering in a cell, it tends to be a bit weaker and less competitive than its natural cousins out there. And so a lot of times you actually have to fight like heck to make a design that will be even viable in the field for weeks at a time. And so to that end, we've worked with our partners to think about identifying naturally occurring microbial isolates that are able to form stable either spores or sort of extracts and isolates that you can more safely apply and control. In other cases, you can think about having different sort of molecular keys or other things where the cell will only have a growth advantage in a very specific environment. But again, that's not foolproof and you have to be very careful. But in general, finding microbial species that very specifically interact with only specific plant or root types is one strategy that folks take. Another is, again, to think about how you can make a either weakened cell or a sort of, spray dried extract. So like half of our ag programs actually have sort of lipopeptide or protein formulations where the cell is no longer around. And so that's another sort of early strategy that should win, approval and deployment. And, there are some products like that on the market too, from the bigger ag players. So something that we'll continue to build around there as well.
Thanks, Nate. A question I received in the chat. If you could comment on how some of your approaches are contributing to reducing greenhouse gas emissions from ag systems.
Yeah, so I guess two main areas there. So for one, we are working in finding microbes that naturally have nitrogen fixation capabilities and then enhancing those pathways so that we can replace nitrogen fertilizer, made by chemical means by the Haber-Bosch process with a greener alternative where the cell itself is able to carry out metabolic activity to provide, ammonia or nitrogen-based fertilizer to the cell. As you guys know better than me, Haber-Bosch is a nasty process and contributes to, the greenhouse gases and pollution quite significantly. Additionally, another field of interest related is thinking about carbon sequestration and thinking about how you can engineer microbes and plant cells to help capture carbon in the soil in a way that it is going to be almost permanently trapped there and or trapped in some other sort of format, but that's like a little further down the road, I think. But yeah, in general, thinking about how to like pull CO2 out of the air and yeah, sequester that carbon is something we're actively working on as well.
Thank you. Ching-An, do any of your students have any specific questions, or are they interested in looking at a particular case study here?
No, I mean, I think student already, they are basically interested to see the case study.
Okay, so any specific case study that one of your students is
I think the first four that's kind of related to the course topics, which they definitely
First four? Okay, yeah, I'll dive in. So yeah, so the way that we think about enzyme discovery, so I mentioned that over the years we've been refining computational algorithms that can take sequence data and try to infer whether or not a new gene sequence may be producing an enzyme that has the activity that you desire. And so what you're looking at here is a very simple sort of sequence distance out diagram where like each node represents a sequence, and the closer they are together, the more similar they are in sequence. What we have done over the last 8 or so years at Ginkgo is built what we call a sort of metagenomic sourcing algorithm, where we'll take either publicly available sequencing data or sequencing data that we might generate from, say, an environmental sample of relevance, and then run our algorithms on that data to think about what enzymes or first the starting gene sequence might have an activity. What I'll say is that this pipeline works extremely well. I continue to be surprised by it, and that you may read a paper or a patent and know that a single gene is known to express an enzyme that gives you the biochemical activity you want. Typically, we'll sample very heavily around those nodes, if you will, saying take a bunch of closely related sequences that might have an activity. But then we also cast a broad net and usually order an extra, 500 to 2,000 putative gene sequences that might have an activity, and then generate real data around them, and then do sort of iterative design build test cycles to look at, sort of new clusters that have activity. Oftentimes, when we apply this, we will find new hits. So, again, this is kind of the scope and scale we work on. And, typically we're running these sort of like low to mid thousands of variants, where sequence homology can be as low as 30% from your starting seed, yet still oftentimes have activity. And so, we'll get these efficiency gains. We'll provide our partners the ability to have freedom to operate because nobody's ever looked at that gene sequence before. And then the other nice thing is you're not looking at the same enzyme sequence that 10 of our collaborating labs have been looking at for a decade and have been engineering to high heaven to get an improved activity. You oftentimes will get a new template that is a better candidate for improvement by protein engineering. And so, what we've been doing now over the years is saying, like, okay, in the context of these different sort of industrial biochemical activities that we need, can we again, think about looking at sequence space and, having different cues in our search algorithms to say, not only do you have to look for like straight homology within domains, but, indication of conservation of residues across places. If you can feed structural data, which is becoming increasingly more accessible. For us, that was a huge challenge because these weren't therapeutic proteins or enzymes, so nobody was getting their structures when that was very expensive and hard. You can infer even with like loose structural data where there could be commonalities. And so in general, continue to sort of refine that search algorithm to be able to find better starting points. And then once we've found those starting points, so for instance, like this is an example, back to Raj's question on like cell as product. Increasingly so these are, being looked at in different therapeutic applications. We partner with a company called Synlogic in the Cambridge, Boston area that is looking at making, sort of microbial solutions for different metabolic syndromes, as well as for cancers and immune relevant deficiencies or conditions. And so in this case, we did sort of a coupled round of metagenomic discovery and then improvement by protein engineering to be able to find a 3 enzyme sub-pathway to be able to look at providing better leucine degradation for patients who have that metabolic deficiency and will build up that metabolite. The idea being that you can engineer these pathways into a cell like E. coli Nissl, which is a naturally occurring microbe in your gut. You can take a pill of that sort of engineered Nissl strain every day and hopefully add increased metabolic activity to work on avoiding buildup of what can be a toxic metabolite. And so doing that sort of iterative combination of discovery improvements and followed on screening, we were able to engineer a new strain with Synologic that was able to drop leucine levels by an additional 7-fold from their best in class strain to hopefully now get into clinical studies. And so yeah, an example of cell as product coupled with enzyme and pathway discovery and optimization. I shouldn't say discovery, we know what the pathway is, but optimization. When we think like, good metabolic engineers and how challenging it can be to make like very large multi-step pathways, you do still have to figure out how to discover that best candidate enzyme. But when you're thinking about flux across a pathway, you really need to balance it well and know that you're generally creating a new bottleneck every time you generate a new cell design. So it's like playing whack-a-mole. You attack one, you create another. And so, we think about both just general rate and activity across a pathway, as well as specificity with our different metabolomic pipelines. And importantly, sort of couple that throughput and bandwidth to test so that we can look at combinatorial pathway designs. And then when we hit a ceiling, we will add on a complementary approach to try to improve that. And so, in this case, another 3-step portion of a pathway that ultimately ended up being in around a 10-step pathway. But in each case, we took sort of low thousands of pathway candidates as starting points, and then we're able to mix and match the best versions of those modules to be able to, and 109 million bases of DNA. Again, scale not for the sake of it, but when you can look at that sort of diversity in combinatorial pathway design, you typically will find winners that you wouldn't have found by a sort of smaller scale hypothesis generation and test. We're able to very significantly, like by more than 18 or 19-fold increase the titer of ultimately 25-fold improvements related here for plant-derived compounds that we work with increasingly. So this is like the timeline of improvement of production of a cannabinoid molecule, a rare cannabinoid molecule that would be impossible to make economically in a plant. So there's hundreds of trace cannabinoids that could have either recreational or therapeutic sort of activities, but plants have obviously, for reasons we appreciate, been evolved to make a lot of THC or CBD, and there's a lot of interesting molecules that you could access. This was I think over 15 to 20 step pathway, starting with sort of all the key components to make all the terpenoid backbones. And you see this, this project took us 3 years to get to work. So the next one, because of code base being generated and understanding, takes you 1.5 years, and the next one takes you 9 months. But there was a lot of work along the way to get up to commercially viable titers by a combination of sort of targeted pathway builds, protein engineering campaigns, and then a lot of design of experiments to improve sort of fermentation process parameters, very specific actually to each new cell design. And then, at the end of the day, when you look at that and you say, okay, I need to get to goal, this is where we typically end up. So there's a 50,000 liter tank at a contract manufacturing organization down in Mexico City that we work with a lot. What we have done over time is, figured out how to be better and better process engineers to be able to build out that infrastructure, but then also take the data that we generate at this scale and translate it back to our automated small scale fermenters so that we can more efficiently bring about process optimization that will still be in effect at scale. We have around 10 deployed commercial organisms and products now running between 50,000 and 200,000 liters. And with the learnings that we have in different, bacterial and fungal systems, are often able to take that small scale optimization where we can test more variables in concert and run good design of experiments with well-powered experiments and say, okay, this is the critical parameter that we need to improve to get better performance in a big tank. Another thing that happens a lot, so looking at this big tank, so for instance, like we do a lot of work in Pichia pastoris. So, a well understood industrial yeast that is able to pretty productively make protein products for you and secrete them. The problem with Pichia is that the canonical industrial strains get turned on by methanol induction. And no CMO wants to fill this tank next to your fermenter with 50,000 liters of methanol. Obviously has safety risks as an explosive solvent. And so what we did to make a more viable industrial yeast host was go on a synthetic promoter hunt where we took some of those sort of multiplexed approaches that I told you about to be able to make a synthetic promoter circuit and then test a number of different combinations of the sort of components of that. So here's a good example of that. So doing a sort of three-way combinatorial assembly, what we were able to do is stack different combinations of those regulatory elements in that promoter system and do it in a simple yet elegant way where upstream of that design, due to the assembly scheme, you would get a stacking of molecular barcodes. And so by doing that, if you could read a sequence and get that readout, you would know which promoters were turning on. And so you could kind of think in terms of your stages of fermentation, how you could control, sort of tight process where you've built up biomass and you can get very productive protein expression and production versus leaky promoters versus weak promoters. And so in general, this is now translated into an industrial Pichia strain that we think is 10 to 20x more efficient and safer than a lot of the industrial strains are out there by, again, testing over 300,000 of these combinatorial designs directly pooled within a fermenter. And so, yeah, using that to, deliver sort of food product. You can think about, different kinds of milk proteins, the heme that, you have in your Impossible Burger, for instance. In general, the food protein space is getting a lot of attention from biotechnology applications and is important to consider. I talked about the COVID-19 stuff. The one thing that we did do similarly upstream of that was address the fact that, and I'll mention verbally, so at the very beginning of the pandemic, we donated quite a bit of in-kind services to partners in biotech and worked directly with Moderna to improve their plasmid production process for making their RNA template for their vaccine. We were able to give sort of vast capacity, so half of our fleet of automated Amber 250 systems to run a 250 fermenter 4-phase design of experiments that resulted in a many-fold improvement in plasmid production for them. Having cracked that nut, everybody was seeing the writing on the wall that the processing enzymes needed to finish off the manufacturing of the vaccines were going to become the next rate limiting step. And so we partnered with Aldevron, who I assume many of you are familiar with, make a lot of valuable enzyme reagents in biotech, to think about how we could improve the design and expression of the vaccinia capping enzyme, one of the two enzymes that finishes off production of your COVID vaccine. And so by looking at sort of different expression systems and subunits, and taking the best design that gave a 5-fold increase in titer, but then coupling that with automated fermentation to improve process, we delivered a process to Aldevron that gave over 10x tighter improvements, which was a very valuable target. So basically gave them enough material so that they could deploy that and be able to satisfy all of their manufacturing partners. So, a good example there. And then I think the last thing I have, we talked about distributing, sort of COVID testing, and I hadn't shown this before when Maxine asked her question, but sort of testing around the world now for variant sequences and different pathogens. But getting more into the immunotherapy and cell therapy arena. In general, I think we're still in the early days when we think about how folks design CAR receptors and think about finding specific T-cell therapies. And there's a lot of really good work being published over the last few years showing, anecdotal evidence that if you could approach the engineering of different domains of the CAR or TCR receptor, that you can make better T-cell therapies. And so we have a number of burgeoning partnerships, some that I cannot talk about, some that I can. So for instance, in vector engineering, we have some work going on with Biogen. Also looking at how we generally try to think about applying the sort of Ginkgo approach of saying, hey, these receptor designs, like these are modular subunits that you can sort of mix and match, evolve via different protein engineering approaches and say, hey, look, we now have the pipeline to be able to look at thousands of receptor designs in the context of a starting T-cell candidate that you might take from a patient. And so, yeah, in general, seeing some interesting data there, as well as, just a lot of really amazing work done in various academic labs, hospitals, and new startups that are coming up in sort of this next wave of cell therapy. So something that we hope to continue to grow our capabilities in. So yeah, that's what I had for you today. Happy to take more questions now, and yeah, good to be involved in this, and thanks again for the invite.
Thank you so much, Nate. Any questions from? Let's see. Okay, I'll ask the final question. So, in the context of metabolic activity and metabolism in general, interorganal communications, right? So specifically, I'm focused on the nuclear mitochondrial crosstalk.
Yeah.
So I was wondering if you could talk about opportunities or approaches that are being taken to target the mitochondrial genome or the mitochondria in general.
I will freely admit that we have not done a lot of mitochondrial targeting, and so I don't have anything intelligent to tell you there. I do know that one thing that we have looked at is more broadly targeting sort of, proteins that you can fuse to an enzyme of interest to be able to anchor it in different organelles. And we have certainly found improvement of, different like P450 CPR enzyme systems and the like, if you target them to the ER, for instance, in certain hosts. And so yeah, we do have that tool set where usually that's maybe like the third or fourth thing you do if things aren't working well, to be like, oh man, I think I need to localize this in the cell. That said, I think, obviously looking at like regulation and gene expression of mammalian hosts, increasingly relevant and important there. And one thing that I'm pretty excited about lately is looking at expression of complex metabolites in plant cells. And they're just like the exquisitely complex sort of zonation within organelles in a plant cell is really powerful to make some stuff that microbes cannot make. And so yeah, early days for us, but we have been playing around with like how to scale plant cell culture for that very reason. Really lets you express pathways that are often just incredibly toxic in your canonical microbial hosts. So worth putting the energy into doing like a new type of cell and host onboarding and try to scale it on the platform.
Thank you, Nate.
My pleasure.
Excuse me, we have a question here.
Yes, Chingan, please go ahead.
Yes, and so thank you, Sarah, for the talk. May I ask if by chance your group has looked into doing any investigative work using your technology to investigate research in the spaceflight field, either with NASA, the ESA, Canada Space Agency, or JAXA? Over.
Yeah, we have thought about it, but I would say nothing of import or significance. I know actually just before the pandemic, a couple of our scientists held a conference at Ginkgo with some different groups about this on, what would it look like for like finding synthetic biology enabled solutions to, get to Mars. I know that there's increasing interest there and, different government programs, but yeah, I think maybe the most real thing that we've considered in writing proposals for is trying to look at different microbial species that could, get you like the starting substrates for, making food in space or, sort of like other applications like that. But yeah, beyond that, we're fairly naive there, I'll admit.
Roger, because it seems like a lot of the cellular engineering and the bioengineering processes they use could be very useful in terms, like you mentioned, food processing, but also in terms like life support systems, perhaps like crossover with the work you're doing in the environmental field, using your reengineering purposes could also be useful for life support systems on spacecraft and whatnot.
I agree. Yeah, no, yeah, something that I hope continues to grow. And one thing of relevance here that I have seen is that with different engineered microbial systems, you can definitely evolve a cell to recognize, some non-traditional substrate or waste stream, and then, turn that waste stream into something useful. And so, we're largely looking at that from like a sort of, mitigation of pollution and or like ag related application area today, but yeah, thinking about what you could do and the constraints that you're dealing with in space is it should play there for sure.
Roger, thank you very much.
Thank you. So yes, Maxine.
How challenging is it from a data storage point of view to keep track of all the different microbes you're working with and the different pathways and metadata that you mentioned? It seems huge.
It's very challenging. I swear that Maxine didn't see my slides, but the slides that I hid are of relevance here. So I mentioned how many software developers we have at Ginkgo. We have a custom LIMS or laboratory information management system environment that is able to track the contents and history and metadata of every sample that we generate. So like here, you're looking at the top row of a multi-well plate. We know who created it, when it was created, what strain lineage is in that row, what data was associated with it. So we have like a software developers like to put LYs in the names, as you guys know. Servicely is as a Ginkgo software tool that lets you, queue your sequencing run and, append your data to that set of tracked and managed samples. All of our samples are barcoded. I talked about DNA barcodes before, but, we also have physical barcodes on all of our plates and tubes so that you can effectively do your work at Ginkgo without having a lab notebook. So the idea is that, you can essentially track that history and track thousands of samples. We actually just had our 10 millionth container at Ginkgo and everybody was trying to compete to get it last month and, trying to run their workflow. And that goes all the way up to, when I talked about robotic systems and work cells, having different kinds of sort of UIs where people can drag protocol modules to be able to track that, and then ultimately look more sort of from like a process optimization mindset to look at how a scheduling algorithm can tell you how a robot should run everything effectively, and then all that metadata and how that run was executed on the robot gets appended back to the sample. And so that's all well and good. It's still not as easy as that. So we have those tools, but we still bury ourselves in data. Around a year and a half ago, we made it like, in addition to all those software and hardware and automation people, we have a dedicated data science team of 20 people today who are like building that next layer of infrastructure over our data so that we can make better reuse of our code base. And, actually just before COVID, we successfully begged the city to be able to run redundant fibers to the Markley data center in downtown Boston, basically emptied our server rooms. So our server rooms are now about 5% as full as they were 3 years ago because just everything goes to the cloud. And that lets us, more efficiently run computation on things, as well as have like backup of data and the like. And so, trying to make those investments, but you can very quickly bury yourself in data.
Okay, I think in the interest of time, we will stop here. Again, I think Nate's presentation has clearly shown the enormous possibilities that exist. We had really good discussions on different possibilities. Shows that it's not Earth is not the limit. Opportunities for going elsewhere as well. So I think it's just fantastic to hear from you, Nate, and we really like to take this opportunity to thank you again for kicking off the Industry Connections webinar series. So thank you once again.
Thank
you
you
all for joining. Yes.
Thanks for having me, and I hope the series goes well.
Okay.
Bye.
Bye, everyone.
Thank you.